Well, if you are traveling, some immigration authorities may require you to go for a COVID-19 test prior to arrival. Essentially, there are two types of tests for COVID-19. Firstly, there are the viral tests, which test for the antigen, and the antibody tests. A viral test will tell you whether you have a current infection, whereby an antibody test tells you whether you've had a previous infection. As you can see, there's a time period for testing the viral antigen, which is within the first week of infection. It's the viral or the RNA test is not good for studying past infections, but it's good for studying or detecting current infections. Whereby the antibody test, whereby you take a blood, it's only good for past infections because antibody does not appear into the blood for COVID-19 until about a week to 3 weeks after infection. Well, this is the blood test for the antibody tests. This is a newspaper article in the Borneo Post on June the 3rd, and it reads, the State Health Department has recommended Malaysians who intend to enter Sabah to undergo reverse transcription polymerase chain reaction or RT PCR test for COVID-19 prior to arriving in the state. So this is a viral RNA test for current infections. And this is from the Ministry of Health Malaysia website. To date, real-time reverse transcription polymerase chain reaction or RT PCR technology is the technology recognized by the WHO, the US CDC, and the Ministry of Health Malaysia for confirmation of COVID-19. So apparently this test is the gold standard test for detection of current COVID-19 infection. This is a diagram of the COVID-19 virus. The things that are protruding out are spike proteins, which help the virus attach itself onto the lung cells or the pneumocytes. The round layer is the lipid layer. That's why health authorities have recommended that you use alcohol or soap and detergent to clean off any potential viral contamination on your palms. And the yellow bit is the RNA material, the brains of the virus, so to speak. RNA is a single strand of nucleic or genetic material, whereby DNA, which is present in all human cells, is a double-stranded molecule. Endogenous RNA obviously has its cellular functions, but an external source of RNA like the coronavirus or COVID-19 attaches itself to our cellular DNA and uses our DNA to produce more copies of itself. And that's how it uses the body cells or the DNA of the cell to spread throughout the whole body. The RT PCR test, which is the gold standard test for current infection, uses a similar method of replication. Firstly, the doctor will put a swab down your nose. It's called a nasopharyngeal swab, which you'll see in a video after this, and also a swab down your throat called pharyngeal swab to try to get cellular material which may contain the virus. And then this is sent to the lab, whereby the RNA is isolated and separated from all the cellular material, the proteins and the fat. And once they've isolated the RNA of the virus, they use a process called reverse transcription to turn it into viral DNA. And then they add complementary parts of DNA to this viral DNA so that when you test it, it can be detected on the machine. And once this is done, they put it into a machine called PCR to amplify it, to make billions of these copies of DNA so that it can be studied in detail and so that they can determine whether you are positive for COVID-19 or not. And this process is called polymerase chain reaction. This is the Star newspaper quoting the Sabah chief minister. If you're visiting Sabah for work or leisure, make sure you have a RT PCR COVID-19 test done prior to arrival and show it at the immigration. If you have a negative test, you do not need to be quarantined for 14 days in the state. Otherwise, you will be quarantined for 14 days. So, I went to a nearby clinic to get my RT PCR COVID-19 test done.
So just pharyngeal swab, nasopharynx.
Yeah, nasopharynx also.
Okay.
Okay. Yeah. Okay.
Done.
Done.
And thankfully, my results were negative. And so stay safe, guys.